to the IXICO webinar, Advancing Alzheimer's Research, Enhancing Clinical Trial Diversity and Relevant Imaging Considerations. The agenda for today, after we do the introductions, we'll have a presentation by Richard Mohs, followed by Robin Wolz, and then a Q&A with Marina Papuzzi as the moderator. So my name is Jane Antique. I am Marketing Communications here at IXICO, and I'll be your host for today's session, and I'll be introducing your presenters for today. So before we get into it, our first presenter here, I have the honor of introducing Richard Mohs, Chief Science Officer at the Global Alzheimer's Platform Foundation. Richard worked at Eli Lilly and Company in several leadership positions, including VP of Neuroscience, Early Clinical Development, and leader of the Global Alzheimer's Drug Development Team. He spent 23 years with the Mount Sinai School of Medicine as a professor in the Department of Psychiatry and as Associate Chief of Staff for the research at the Bronx Veterans Affairs Medical Center. He has a number of accolades across the board here, so I'm not going to go through them all in detail, but of course, I'm looking forward to seeing his presentation as well. Joining Richard will also be Robin Wolz, Chief Scientific Officer at IXICO. Robin's had over 15 years of experience in development of innovative analytic solutions in healthcare with a focus on imaging technology. And prior to IXICO, Robin held different roles at Philips in the research and diagnostics X-ray divisions. And of course, Robin holds a PhD in medical imaging and computer science from Imperial College University and focused on early detection of Alzheimer's disease. And then our moderator, Marina Papuzzi, she is a senior biomarker scientist here at IXICO, and she's got more than 15 years of experience in neuroimaging for CNS in both academia and industry. And she currently supports many of our clients and studies which use medical imaging for movement disorders such as Huntington's, ataxias, and Parkinsonian syndromes. So welcome aboard, team. I'm looking forward to all your presentations. And I think without further ado, I'm going to pass it over to Richard for his presentation. And then afterwards, I'll then transition over and hand over to Robin once Richard's finished. Over to you then, Richard.
Okay. Thank you, Jane. If we could have my first slide. I'm going to talk today about some work that I've been doing with the Global Alzheimer's Platform Foundation along with a number of partners that we have in the pharmaceutical and biomarker industry. To try to gather data that would help us understand better the utility of both blood-based and digital biomarkers in the context of current clinical trials. Next slide, please. I think the next slide will show my disclosures. There we go. Nothing particularly remarkable there, but I'm speaking on behalf. Next, this gives you some idea of the current developments. It's been a very productive past few years for Alzheimer's disease research with developments that are going to have a major impact, not only for researchers, but also for patients and for clinicians who treat patients with Alzheimer's disease. Most important is that we have now some disease-modifying monoclonal antibodies that have been shown to actually slow the progression of disease. This is really a major first, and they work by removing amyloid plaques primarily. And the development of these compounds has been possible only because of the development of amyloid PET technology that allows us in life to measure the density of amyloid plaques. And without that technology, these drugs would have been virtually impossible to develop. So along with that development in therapeutics and PET technology, we now have remarkably, for somebody like me who's been looking at CNS diseases for a long time, we have some biomarkers that one can measure in blood, which seem to be a pretty good reflection of the degree of pathology in brain. And those are still under development. We don't understand them completely, but things like A beta 42/40 ratio and some of the p-tau species seem to be very good reflections of the degree of pathology in brain. And it's quite clear that as we further develop therapeutics that are based on reduction of amyloid or reduction at A beta or the removal of tau, it will be very important to use biomarkers, hopefully primarily blood-based biomarkers, to select patients and then to gauge the pharmacodynamic effects of those treatments. So next slide. The difficulty, of course, that sponsors have in doing these trials to enable them to evaluate patients is that, first of all, you have to pick people who are at a certain level of a symptom or sign severity and usually sponsors either look for people who are cognitively normal, or most of the trials now look for patients who have mild cognitive impairment, that is a memory impairment that looks like Alzheimer's disease, but they are not yet functionally impaired. Or then when you get uh, uh, additional functional impairment, in other words, impairment in actual activities of daily living, we would then say that that's likely to be mild AD. Now, screening of large numbers of people for these conditions with amyloid PET or CSF is possible, but it's time consuming and not really very practical. Um, the advent of better blood-based and digital biomarkers uh, looks like we could be able to, in some cases, eliminate that step in the process. But most of the work that has been done to date on these validating these biomarkers has used convenience samples. In other words, they've been validated and looked at in studies that were selected or that were developed for other purposes, such as to understand the genetics of Alzheimer's disease, certain people that were followed for autopsy studies or epidemiologic studies. And from the point of view of a drug developer, it's a concern that these kinds of samples might not directly mimic the sort of patients who are recruited for clinical trials. Additionally, particularly for studies done in the US, clinical trials, it is expected now, are likely to enroll populations that are representative of the US population, and that would include a representation of people who are Black, African American, or Hispanic, as well as white folk, as well as white people. And many of the studies that have been done to look at blood-based and digital biomarkers to date have not really done that. So that was one of the aims of the Bio-Hermes study. Can we have the next slide, please? This just shows probably the genesis of the concern about the mix of people who are currently in clinical trials. This looks at the phase 3 trials that were done for solanezumab, aducanumab, lecanemab, and donanemab. And you see that at least for people who were recruited in the US into those trials, the vast majority were white. And the representation, particularly Black African Americans, was quite small. Some of those trials did enroll a fair number of people of Asian background, and that's because they're done globally. But in the US, that representation has not been very high. In the lecanemab studies, there was a reasonable representation of Hispanics, but clearly this is a concern as we move forward to try to get a better understanding of how these treatments and blood-based biomarkers might perform among people who are not highly educated white people. So next slide. Okay, the blood-based and digital biomarkers that were included in the Bio-Hermes study included a broad range of blood-based biomarkers, many of which we have not fully analyzed yet. We also have some digital biomarkers that might someday reflect amyloid pathology in brain. We haven't fully analyzed those yet. What we're going to look at today is primarily the major blood-based biomarkers, A beta and the p-tau species and their relationship to amyloid. Now, it's important to notice that when we look at these blood-based biomarkers from a regulatory point of view, we have to define a context of use. In other words, what does the clinical data show that they might be good for? And I think that the Bio-Hermes data are relevant, at least, to the assessment of these biomarkers as susceptibility or risk biomarkers, possibly have some relevance for diagnosis and prognosis in terms of their ability to predict brain pathology. We do not have longitudinal data, and this is not a therapeutic trial, so they're not relevant to the use of biomarkers for predicting progression or for measurement of pharmacodynamics. So could we go to the next slide, please? This is the way the Bio-Hermes study enrolled people. We were interested in people in the clinical groups that drug developers are typically interested in. We are interested in people who are either cognitively normal, have mild cognitive impairment, or mild AD. We over-enrolled in the cognitively normal group, and that was done by design because we wanted to have enough people who were cognitively normal but had some degree of amyloid pathology on PET so that we could assess the utility of our blood-based biomarkers for predicting amyloid PET positivity in this group where the rate of amyloid positivity is low. You'll notice also that we were quite good at recruiting members of underrepresented populations. In the US, that's Blacks and Hispanic folks, and we had a number of those who were represented in each of the clinical groups, that is the cognitively normal, MCI, or mild AD patients. At one of our sites, we did not have access to amyloid PET and had to use CSF, but that's a very small number of people, and I won't really be discussing them much in this presentation. So next slide, please. This shows the overall rate of amyloid PET positivity done by visual read in the 3 clinical cohorts. As expected, we see that the rate of amyloid positivity is fairly low among people who are cognitively normal. It's 21%. Now, it might have been even lower than that, but we did exclude people who were very, very good on their initial cognitive screen because there's data that shows that those people have such a low likelihood of being amyloid PET positive that people who were more than 2 standard deviations above normal for their age group were excluded from the study. But so we got a low level there. The rate of amyloid positivity goes up among those who have a memory impairment but are not functionally impaired, that's MCI. And then of course it's much higher among people who have the clinical features of mild AD. Next slide. Okay, this just shows the basic demographics of the people in these different groups. I won't go through everything in detail, but the important thing here is to look at what differentiates people who are amyloid positive versus those who are negative. In general, the people who are amyloid positive are significantly older. They do not differ based on gender. In other words, there's no difference in the rate of amyloid positivity for men or women, or at least the number of men and women did not differ by those groups. Education did not differ significantly for people who are amyloid PET positive versus negative. The level of cognitive impairment, as we would expect, was greater among people who were amyloid PET positive. Their MMSE scores were lower. And their RAVALT. RAVALT is the Ray Auditory Verbal Learning Test. That's the memory test that was used to screen people into the MCI group. That performance is lower among people who are amyloid PET positive, and they had more functional impairment. The FAQ is a functional activities questionnaire. It's a measure of activities of daily living. And as we would expect, the proportion of people who were carriers of an ApoE4 allele was markedly higher among people who were amyloid PET positive. So no effect of gender or education, but the other variables fall out the way we would expect from previous series. And by the way, the demographics of this group of people is very similar to what are typically enrolled in current therapeutic trials of new disease-modifying drugs. Next slide, please. Very interestingly and consistent with what I think many sponsors have found, if you just, because we use the same clinical criteria for enrolling people regardless of what their racial or ethnic background was. In other words, we were looking for the same signs and symptoms of disease, and we had the same medical rule-outs, et cetera. But the overall rate of amyloid positivity was just higher among non-Hispanic whites and to some extent among Hispanics than it was among non-Hispanic Black participants. It was just lower. And this is consistent with data, for example, from other studies where even if you just go out and look for the same clinical phenotypes, the rate of amyloid PET positivity among non-Hispanic Blacks just usually turns out to be lower. This is true for example, in the IDEAS study that was conducted in the US. We don't really understand that very well, but it simply is a phenomenon that drug developers have to deal with, and we found it in the Bio-Hermes study. So next slide, please. This shows the potential differences overall, regardless of clinical state, for the different racial and ethnic groups. And here we find that there were slight differences in mean age. The Hispanics were and African Americans were somewhat younger than the white participants. The ratio of the number of females among African Americans was much higher. Conversely, the number of males recruited among African Americans was much higher than it was for either Hispanics or non-Hispanic whites. There was small differences in average educational background, although the average education level was quite high for all racial and ethnic groups, but it was slightly higher among non-Hispanic whites than the other groups. There was no difference in average MMSE score. I think we'll have to dismiss here and not make anything of the Rayvault scores because we use different norms for the different racial and ethnic groups on the Rayvault, so these scores are not really comparable there. And we shouldn't do that. But the overall measure of cognition for the MMSE, for example, was not different. The FAQ was not significantly different. And interestingly, the proportion of ApoE4 carriers was not different. So on an overall basis, demographically, there really wasn't a huge difference among the racial and ethnic groups. Next slide, please. This shows, I won't go through the details of how we calculated, but the key question that we were interested in is what's the relationship between each of these blood-based biomarkers and our gold standard, if you will, which is visual read of amyloid PET positivity. And we see there the blue line is represents p-tau217, which is a blood-based biomarker. And it consistently, if you go to the upper left-hand corner there, both the sensitivity, in other words, the ability of the test to pick out people who are truly amyloid PET positive, and the specificity, that is the ability of the test to show who is not amyloid PET positive, was higher for p-tau217 than for either of the other two widely talked about blood-based biomarkers, that is p-tau 181 and the A-beta 42 to 40 ratio. Now, the A-beta 42 to 40 and p-tau 181 blood-based biomarkers are actually not bad. They are quite predictive of amyloid PET positivity. They just aren't quite as good. Now, as I mentioned earlier, the overall rate of amyloid positivity was lower among non-Hispanic blacks than it was among non-Hispanic whites, and slightly lower than among non-Hispanic than among Hispanics. But if you look at the ability of the test, the blood-based tests, to predict amyloid positivity, it was really quite similar across all of these groups. So although the base rates are different, the ability of the test to predict was not significantly different based on race or ethnicity. You see the lines are a little bit choppier there for non-Hispanic blacks and Hispanics, and that's simply because we have smaller numbers of people there. You also notice that the ordering of the predictive power of the blood-based biomarkers is the same, so that p-tau217 is better, slightly better, but significantly better than A-beta 42 to 40 ratio or p-tau 181 for all racial and ethnic groups. And so I think this gives us confidence in the ability of p-tau217 to predict who is likely to be amyloid PET positivity, regardless of racial or ethnic background. It does not explain why the overall rate of amyloid PET positivity is lower among blacks than it is among the two other groups. But the technical performance of the test is quite similar for the different groups. Next slide, please. Okay, so here's a summary of what I can say about these blood-based biomarkers from Bio-Hermes. We know that they have similar age to the clinical trial participants. This is comforting for people who are designing clinical trials. Amyloid positivity was associated across all groups with greater cognitive and functional impairment, older age, higher rate of APOE4 carriers, and it was higher among whites and Hispanics than among blacks. The last finding we really don't have a great explanation for, but we'll continue to look into that. Amyloid PET positivity was not associated with either gender or education. And the blood-based biomarkers in this set of folks who are very similar to the kind who would be enrolled in clinical trials showed that all of these blood-based biomarkers that we looked at here were quite good, but p-tau 217 was consistently the best for predicting amyloid PET positivity. And as far as the technical performance of the test, it was not different by race and ethnicity. Next slide. People often wonder, because the rate of inclusion of underrepresented minorities is often so low, at least up to the present time in clinical trials, how did GAP do such a good job at enrolling these folks? Well, we did several things, and we used both the carrot and the stick. The carrot was that GAP provided group people who were called community connectors. They were GAP staff, most of whom are from underrepresented minorities themselves, helped our sites in GAP to establish ongoing relationships with minority-led community organizations. In certain areas, particularly Florida and the US, GAP had mobile units that were provided by GAP to enable screening in communities that eliminated the need for people in these communities to travel to clinical trial sites outside of their community. These vans were equipped to do all the clinical screening necessary to get people started in the trial. GAP also established IRI, we call it Inclusive Research Initiative Advisory Committees, which were comprised of leaders from minority communities that met with GAP and site staff to get them apprised of what the study was like, make them feel comfortable with it, help to answer their questions, etc. And so these were the kinds of community outreach efforts that GAP engaged with to enable sites to at least come in contact with a much higher proportion of people from underrepresented minorities. Now, we also did put in a little bit of a stick with the sites, and that is that sites were required, after they had enrolled a certain number of non-Hispanic whites, that in order to continue in the study, they had to make a real effort to increase the enrollment of members, participants from underrepresented minority communities. And the last point I would emphasize is it helps to very much, I think, to have site staff and GAP staff who were members from these underrepresented minorities, either Hispanic or Black people. So it can be done, and I think that sponsors should be encouraged to use these kinds of activities to up their enrollment so that the people enrolled in clinical trials used to register these new treatments are actually representative of the entire population that is likely to be treated. I think that's the last slide, Jane. So I will thank you, and moving on to Robin.
And thank you very much for that presentation there, Richard. It was super insightful. And I think that it's given a lot of people, a lot of food for thought as to how we can approach different types of different ways of clinical trial design, and also implementing the various tactics that GAP have done to increase the number of underrepresented minorities into the study. So thank you for that, Richard. Very enlightful. Robin, I now pass over to you for your presentation.
Thanks, Jane. Thanks, Richard. Good morning, everyone. Good afternoon, depending on where you are. So as the slide says, I'll talk a little bit about potential reflections and considerations on what the different points may mean from an imaging point of view. So if you go to the next slide, please. Sorry, I don't see. Yeah, there you go. So as Richard, I guess, pointed out in the beginning, amyloid is a key part of the biological definition of Alzheimer's disease. It's well rooted in the ATN framework that has been proposed and developed over the years. It is a key selection criteria across 80 clinical trials, specifically those targeting amyloid, of course, but more generally, a disease-modifying therapy. It's traditionally done through an amyloid PET or a CSF assessment. As Richard said, we see increasing validity or results from blood-based biomarkers, and we see certainly the path towards using blood-based assessment for amyloid at inclusion. The other area that is of course critical is efficacy longitudinally, where amyloid PET is widely deployed, especially again as we are looking at anti-amyloid therapy, and the reduction in amyloid plaque is a key efficacy outcome. It has been considered an endpoint of reasonable likelihood for surrogacy by the FDA in both the Aduhelm and Leqembi approvals. So certainly raising the bar for the role that amyloid PET can play in future clinical trials, also reflected in the recent draft guidance issued by the FDA on AD drug development. So what I want to talk about a little bit, so I'll get back to the PET results from Bio-Hermes, explore that a little bit further, but then specifically with that view on traditionally underrepresented populations, what does it mean for recruitment, what does it mean for interpretation of the results, and I thought it's worth maybe framing that with a quote from the popular press. So in the bottom right here, you see an article on NBC News headline, promising new Alzheimer's drug may be less effective for Black patients. If you then read through this, you come across the sentence, prospective Black volunteers with early disease symptoms did not have enough amyloid in the brain to qualify for the trials. So if you look at this through a more technical or scientific mindset, you will see that there's significantly higher screen failure rates on amyloid on the amyloid step in the Black population compared to white subjects. Obviously, that screening for amyloid will be after screening for clinical severity. So what we've seen on Leqembi, for example, is a 55% screen failure rate on amyloid for Hispanics, 49% for Black subjects, and only 22% for white subjects, all of which have gone through the same screening funnel as far as clinical assessment is concerned. And that then overall led to this significant underrepresentation of those populations in the trial, where only 43 Black participants, for example, got into a trial which represents 4.5%, where the US population, we have 14%. So the key questions that, and obviously there's then follow-on questions from that. So what does that potential difference, or what does that observation mean for stopping criteria in anti-amyloid therapy, i.e. what levels of amyloid you need to get to stop treatment? Is there any consideration that derives from that significant difference in terms of recruitment rates? And then more generally, how do we assess amyloid across diverse trial populations? I'll get a little bit into visual read versus quantitative analysis there. And then more generally, I guess the key questions the field is trying to work through is really, is there a direct link between race and ethnicity and the biomarker assessment? So in this case, amyloid accumulation. So i.e. are the biomarker assessments that are predominantly built on white subjects, so for example, the visual read criteria, do they not generalize well enough to other populations? That's one hypothesis. I think another strong hypothesis that I'll touch on a little bit is whether there are any confounding lifestyle effects as opposed to true differences in terms of the underlying race and ethnicity that then might drive comorbidities that explain the differences in findings. So these are really the topics I want to cover, and before going into those more general questions, I'll come back to the Bio-Hermes results. So if you go to the next slide, I will be recapping some of the data that Richard has shown, but then specifically focus on the PET findings and interpretation of those. So as Richard has shown, Bio-Hermes has recruited 24% of visually underrepresented subjects, 229. There's a slightly higher proportion of 80 subjects in that group. It's not statistically significant here, but it is in line with a higher prevalence of 80 and related disorders in Black and Hispanic groups that has been reported before. We see a slightly higher female proportion in the Black group compared to the white group. And then we see slightly lower MMSE in the Black group compared to the white group, and no significant difference in E4 carrier status. Go to the next slide, please. Which will be now getting into the PET analysis performed on the trial. So a standard flow beta peer amyloid scan was acquired for all subjects. The data was collected in a central imaging platform before undergoing visual QC by a trained expert. SUVR analysis, so quantitative PET analysis, was then performed in the global cortical average using the MIM software package. Again, all of this was done centrally. And then finally, visual read into amyloid positive and negative was performed by a group of central readers trained in the manufacture read process and following the VisQ process. So VisQ here means the expert reader will get presented the global SUVR value and will then make a final judgment or decision on amyloid positivity based on the visual read criteria. Moving on to the next slide, please. Briefly looking at amyloid levels and amyloid rates across the different clinical groups. So we see good balance across age, gender, and education, but as Richard pointed out, slightly higher or slightly lower proportion of females in the 80 group compared to the cognitively normal group, slightly higher age, and then again, education is slightly lower in the mild AD group compared to the cognitively normal group. There are no significant differences in those characteristics when comparing the amyloid positive and amyloid negative groups. Now let's look at the PET results by clinical groups. So as expected, really amyloid positivity on visual read goes up as we go to the more impaired groups with 21, 35, and 60% respectively for cognitively normal MCI and AD groups. And similarly, we see the expected increase in global cortical SUVR as we go to more cognitively impaired groups. Look at the overall population amyloid negatives showing average SUVR of 1.0, whereas amyloid positive on average SUVR of 1.43. Now moving on to the next slide, please, which gives a brief assessment on ApoE4 carrier status. So we're comparing here E4 non-carriers versus subjects that carry at least one allele, so one or two of the E4 alleles. What we can see again is the expected differences with significantly higher amyloid load and positivity rate in the carriers versus non-carriers. We see higher carrier rate in the amyloid positive group compared to a negative group, which is kind of like the same thing looking at from the other side. And then we see, again, as expected, SUVR is higher in the carriers than the non-carriers. Next slide, please. This now gets into the key findings here in terms of the differences in race and ethnicity. So as Richard has already indicated at a high level, the key finding here really is the difference in amyloid positivity level between the non-Hispanic or Latino, Black, and white groups. We don't see significant differences between the Hispanic or Latino or non-Hispanic and Latino ethnic overall groups, and no differences to the smaller, so the other racist group here really. So the key focus is on the white versus the Black, non-Hispanic or Latino, where we see this quite significant difference that Richard also mentioned in terms of 37% positivity in the whites and 26% positivity in the Black subjects. Interestingly, what we see at the very bottom here is that the mean SUVR is not significantly different between those two groups, and only slightly different, or basically 1.16 versus 1.13, and that is something I'll touch on in one of the following slides in a bit more detail. So if you move to the next slide, please. This one basically now zooms in a little bit to the non-Hispanic or Latino group overall, and compares the amyloid negative and amyloid positive groups, and then within those two subgroups, the white and the Black subpopulations. So what we see, if we start with the amyloid negative group, for example, we see that MSE is lower in the Black group compared to the white group. This trend is not significant in the amyloid positive group, which might link to the small sample sizes. But what we then interestingly see is that SUVR is higher in the amyloid negative Black compared to the amyloid negative white group. This is a relatively small difference that is significant though. However, a trend in the opposite direction is not statistically significant in the amyloid positive group. So if we reflect on those findings, what we basically see is that there is this significant difference in overall amyloid positivity rate as determined by the gold standard visual read, but that is not reflected in a mean difference in global SUVR. So there must be some nonlinear patterns or local distribution effects that link to those. And that's indeed something we're looking at the moment, but more generally, to better put those findings into context, what we need to do is look at the relationship between visual read and quantitative amyloid assessment, which we'll do to some extent on the next slide. So if you move on, please. As people might know, there's a quite widely established cutoff to define amyloid positivity from flow beta peer global cortical SUVR, which has been proposed at an SUVR of 1.1. So if we overlay this cutoff in the horizontal line here over the amyloid negative and amyloid positive groups in the Black and white populations of Bio-Hermes, we see some quite interesting trends. So if we look at the amyloid positive fraction, so that's the right hand bar of the Black group on the left and on the white group on the right, we see actually in both groups 7% of subjects that are scored amyloid positive, or that are rated amyloid positive by the visual read, but fall below the line of SUVR 1.1, which means these are cases where there's a discordance between the visual read and this quantitative cutoff of 1.1. So 7% of cases that show relatively low global cortical SUVR in both groups are still rated amyloid positive by the visual read. If we now look at the amyloid negative group for both subjects, we see that discordance is 13% in the white population. So 13% of cases that show SUVR above 1.1 but are still rated amyloid negative. However, this is almost twice the percentage in the Black or African Americans, with 24% that show elevated SUVR but are rated amyloid negative. So that is interesting and is probably the key explanation for this effect we saw on the previous slide, where there is a clear difference in visual read amyloid positivity, but not the mean SUVR. This is something we are currently investigating further by looking at at basically local uptake patterns. So are there any differences in the uptake patterns that we see between the different racial groups that may explain why those subjects are more likely not to be rated amyloid positive, even though they show higher levels of global SUVR. So if you go to the next slide, I'll briefly recap those findings and then try and bring it back to the bigger picture. So what we've seen in those initial PET findings in Bio-Hermes is a low rate of amyloid positivity in the Black or African American group compared to the white group. Global SUVR shows no significant difference between the different ethnic and racial groups assessed. And then we see a higher percentage of subjects in the Black or African American group compared to the white group that show elevated SUVR, do not score positive on visual read. So that's where we are. And the key questions that this leads us to, and that I think are critical for the community more generally, is what I'm trying to summarize here at the bottom part. So one key question is, it's a somewhat simplistic question, but still, do visual read criteria require adjustment to capture homogeneous populations? That's like one hypothesis, which would basically explain that the difference in amyloid uptake patterns between those groups might describe the difference between SUVR and visual read we've seen. But then, of course, there's a key question: what might those differences be linked to if that hypothesis holds? An alternative hypothesis that I think again has a strong support from some of the findings I'll get into on the next slide is: are instead those differences in amyloid positivity rate driven by confounding effects that are not directly linked to race and ethnicity directly? For example, systematic differences in socioeconomic status, which then might drive systematic differences in comorbidities that could explain the fact that we're seeing different levels of amyloid positivity in between subject groups that overall show the same level of clinical impairment. And to motivate those hypotheses in a little bit more detail, I have now 2 or 3 slides to talk a little bit about some relevant literature findings and then try and bring it back to the work we continue to do here. So if you go to the next slide, this is really talking to the first question we've articulated just now, which is there a direct relationship between race and biomarker measurements? i.e., are these differences in PET patterns that we're seeing directly linked to the underlying difference in race? Morris et al. Have investigated this question in a cohort of around 1,200 individuals in a single-site study on natural aging and AD at the Knight Alzheimer's Disease Research Center at Washington University. I'm kind of like quoting directly from the paper here with their top-level findings, which is that there is a possible race-dependent biological mechanism that contributes to expression of disease. However, zooming in to those results, we see that basically this quote comes back to the fact that there was an interaction of race and ApoE4 status that has shown that lower CSF tau was measured in African American who carry at least one E4 allele. There were, however, no racial differences in tau markers for those who did not carry an ApoE4 allele. There were no differences observed in PET. There was, interestingly, a difference in vascular risk factor that was reported, but no difference in cerebral ischemic lesions, which was measured from MRI. So that shows how we're looking at a quite complex picture as we're trying to understand those relationships in more detail. So ultimately, what those findings underline is that in reality, it's very difficult to disentangle the different contributions of different factors to clinically observed dementia that we're seeing here. I'd like to highlight or point out that the fact that at least there seems to be some indication around differences in vascular risk between the different racial groups, even also in this study, which potentially could be driven by systematic differences in lifestyle factors, does support a hypothesis that this could be a potential explanation for the observed biomarker differences. So with that thought, if we go to the next slide, please. I'd like to dive a little bit more into the topic of vascular risk. And there's a recent paper by Daniel et al., which is a study that aims at investigating the underlying reasons for higher prevalence of AD-related disorders in Black and Hispanic populations compared to non-Hispanic whites that has been reported by the US Center for Disease Control and Prevention. So what this study does is it looks at the risk of developing dementia through a defined vulnerability index, which essentially is considering different demographic variables and relevant risk factors. The key results of this study is that education plays an important contribution to vulnerability in this study, where with white non-Hispanics subjects having the highest educational attainment and Hispanics the lowest. So that's an important finding that might be expected. It does, however, not explain what we've seen in the Bio-Hermes trial, as education was largely comparable in the different racial groups. The second key finding from Daniel et al. Is a difference in vascular risk factor again, which describes a significant portion of the overall difference in risk profile to develop dementia. So this finding further supports the hypothesis concluded from the previous paper and discussed throughout the presentation. If you go to the next slide, just the last literature assessment. So overall, but those papers that I've shown, and there are many more papers looking at those topics and other studies. So a strong hypothesis does seem to be that there are systematic differences in comorbidity between the different racial groups that could be a potential or likely contributor to the observed difference in biomarker performance, like amyloid assessment we focused on here. Specifically, potentially increased vascular risk that has been reported in some studies in the Black or African American groups may contribute to the clinical dementia symptoms that are not reflected then in amyloid deposition. And if we bring it back to the screen failure rates we discussed for Leqembi, that may explain why someone qualifies from a clinical assessment point of view, but then fails on amyloid. There's a small twist to this, and to make things a bit more complicated, there's then a related question on whether an increased vascular risk might directly drive amyloid pathology, which of course would discount some of the arguments I just made, but is another line of research. If that's the case, it could potentially explain different patterns in amyloid pathology we see depending on what level of vascular burden the subject has. That's something that, amongst other researchers, was investigated by Rodrigue et al. 10 years ago already. And the finding here was that subjects with hypertension and at least one E4 allele did show significantly greater amyloid burden than those with only one of those risk factors. So we know that E4 status is a key risk factor and key driver for amyloid, but then subjects with hypertension again showed an increased accumulation in this study, supporting a hypothesis that there's a link between vascular risk and amyloid burden as well. So again, these are quite subtle findings and nuances that just show how difficult it is to quantify the interaction between vascular risk, amyloid pathology, how the two then contribute to overall cognitive burden, and how that can be considered in the context of drug development, of course. So I hope that didn't add confusion, but did point to some of the relevant considerations here. But I'd like to wrap it up, go to my last slide, try to bring it back to what it is we're planning to do at IXICO and in our partnership with GAP to understand some of those questions in more detail. So key question is, as I said, is an investigation on more detailed investigation on the different uptake patterns we've seen between racial groups, because that will basically help us understand whether there's a direct link to race, and whether that may require considering difference in reading paradigms, even though I think here the second hypothesis is a much stronger hypothesis based on the literature results, which is really around better understanding vascular risk, quantifying vascular risk, and trying to understand the link to amyloid accumulation and how that has to be considered as part of treatment trials. So the key question is, does midlife vascular risk contribute to AD pathology, or is it an independent contributor to clinical dementia? And I think to understand those questions, we need to better understand vascular risk in natural history studies in Alzheimer's disease, and probably also need to more carefully think about vascular risk as we plan AD clinical trials. So typically, vascular pathology, of course, is a key inclusion criteria in AD trials. So again, most disease-modifying therapy would include some exclusion criteria based on substantial white matter hyperintensity or micropletes above a certain defined threshold. However, there are additional findings that can be taken from an MRI, and that, if analyzed more quantitatively potentially or in a more nuanced way, might provide a more continuous assessment of subject's vascular burden, which helps understand those different relationships we looked at today. So I'd like to close, but briefly refer to the upcoming Bio-Hermes trial, which our partners at GAP are planning in the final stages. And the reason I'd like to point this out here is because, amongst other additions, the Bio-Hermes 2 protocol will include a detailed MRI workup, and will therefore allow to quantitatively assess vascular pathology in more detail, which then will hopefully provide more context to understand these topics in more detail. And that's all for me. I'm probably over time, and hand back to Jane here. Thanks.
No worries there, Robin. I think it was super fascinating to see like the vascular pathology element and the nuances of comorbidities when we're looking at the in the context of Bio-Hermes. So I want to bring back the panel and then also introduce Marina, who's going to dive in straight to the Q&A. I appreciate that we're a little bit over time, but hopefully if people have a little extra 5 minutes to ask questions, that'd be fantastic. Richard and Robin, you can also unmute as well. There is a couple of questions already in the chat, so but Marina, I'll pass it over to you to kick off the Q&A.
Thank you, Jamie. So maybe I'll start with you, Robin, because you made a really nice segue to one of the questions. So you mentioned that vascular risk may be a factor underlying the differences in amyloid pathology between different groups, and that Bio-Hermes 2 will include MRI as a way of measuring vascular pathology. I wonder whether you could comment on how we can use MRI to measure vascular pathology in clinical trials. Thank you.
Yeah, sure. Thanks. I'll keep it brief, and I'm happy to follow up in more detail, just because I think I ran out of time a little bit. So I mentioned white matter hyperintensities and cerebral micropletes, which are quite commonly assessed already in most AD clinical trials. But then there are other MRI findings that can be considered to assess vascular burden. There's actually an established criteria called the STRIVE criteria that has recently been renewed into STRIVE 2 criteria, which is basically around neuroimaging assessment for vascular pathology, and it considers different MRI sequences, including diffusion, structural, SWI sequences to look at various pathologies, including the ones I've mentioned, but then also superficial siderosis, small infarcts, lacunes, and other findings. And I think if we carefully assess those, potentially using quantitative tools as opposed to cruder visual rating scales, we might be able to get a more sensitive signal of vascular burden. Thanks.
Thank you. That's wonderful. I've got one more question, this time for you, Richard. So you spoke about the importance of clear blood-based biomarkers in Alzheimer's disease. And so I'm wondering whether you can comment, what role do you think they will play in maybe clinical trials in the future? And also, how do you see them being used in relation to the existing imaging biomarkers? Thank you.
Yeah, I think we're very fortunate that the blood-based biomarkers have turned out to be as good as they have turned out to be at uh, predicting who is likely to have amyloid pathology uh, if measured by amyloid PET. And given the expense and the limited availability of amyloid PET and so forth, and the fact that the blood-based tests, particularly p-tau 217, are so good, I think for many, many people, not everybody, a p-tau 217 test may be sufficient to determine whether or not they are eligible for or should be enrolled in a clinical trial of an anti-amyloid therapy. I think there will continue to be cases where it is not 100% sure, such as those that Robin mentioned, where you've got a pretty high SUVR, but you're not amyloid positive on a visual read. I think you may need some more granularity there, and you'll continue to have to use amyloid PET scans or other kind of PET scans. And I would hope that over time we can develop blood-based tests that allow us to assess for other kinds of pathology as well, including things like alpha-synuclein, progranulin, maybe vascular, I don't know. But it would be helpful to have blood tests for other things. But I think for now, they are good enough so that in many cases, I would be comfortable enrolling people in a clinical trial on the basis of a blood test, maybe without a follow-up amyloid scan.
Thank you. On that same note, Richard, maybe you can comment on the another question. Are there any plans to replace PET positivity or SUVR with more reliable and predictive outcome measures such as hippocampal or entorhinal atrophy? So these are more clinically relevant and less varied between races. So now this is a completely different perspective, not even
Yeah,
blood-based biomarkers, right?
yeah, I really don't know about that. I don't know that any of those are a great candidate right now. And by the way, I'm not sure, given there was this discrepancy between the way Lilly did their trials and the way Eisai did their trials, where Lilly stopped when people got to a very low level of amyloid deposition in brain, whether or not you could assess that threshold through blood-based biomarkers, I think still remains to be determined. But that's starting treatment is one issue, deciding when to stop when you've done as much good as you can do and you don't want to continue just to do harm. That's something that we need more information about these blood-based tests to determine.
That's great. I think we're running out of time, so I'll pass it on to Jamie. Thank you so much again.
Thanks, Marina, for going through the questions there. We had a fair few, so if we didn't get through to them, we will answer them either in email or in subsequent correspondence. But I just want to say thank you to everybody that's joined us wherever you are in the world. Before you go, don't forget to follow us on LinkedIn and Twitter. There will be a recording of this webinar, which will be sent to you once the webinar is concluded. And if we don't answer your questions today, as mentioned before, we'll reach out to you later on at some point to make sure that we do answer the questions. There is a survey at the end of this webinar, and your insight and feedback is going to be important in helping us curate our future webinars. So if you can spare 5 minutes, please do fill them out. I'd like to thank the panel for joining us and presenting their insight today. It's been very much food for thought for me as well. And if anything else, I want you all to have a very, very fantastic day. Take care, and thank you for joining the webinar. Take care all.
Thank you.